Chapter 24, Part B, Nutrition, Metabolism, and Energy Balance. Okay, let's move it and finish this chapter with the second part. Now, the goal here is to produce molecules that can be used for energy in the citric acid cycle or get converted into glucose. This particular diagram, you've seen this already in Part A, and it pretty much explained to you what the amino acid pool is. So we are just going to get a review so you will understand and keep your mind on track, because the metabolism of the protein is not yet finished. We're going to finish it at the start or the beginning of this chapter, which is just now. Okay, so proteins pretty much would deteriorate, so they need to be continually broken down and replaced. And amino acids are recycled into new proteins or different compounds. Proteins are not stored in the body. Now, when dietary proteins are in excess, amino acids are either oxidized for energy or converted to fat for storage. So those of you who are in a keto diet, and if you are overdoing the keto diet and you think the protein is actually good for you, rather than the carbohydrates, just know that if you are excessively taking in protein, they are not really stored in the body. They will actually be converted into what the amino acid pool is actually converted and oxidized for energy or converted to fat. So you really do get fat also. So if you're on a keto diet, make sure that you burn those proteins, because or else they're going to be converted into glucose. So the first step in the amino acid breakdown, pretty much when you break down protein, is to deaminate. Therefore, the amine group or the NH2 group is removed and then converted into pyruvic acid or one of the keto intermediates of the citric acid cycle. So, having said that, let's take a look at the events of the degradation of amino acids. So, you have a lot of amino acid. You just had a big plate of protein and you got too much. Some of them um, will be used as fat. So how does it go? So the events in degradation, you have to pretty much like chop down the amino acid group that you've taken in from the amino acid pool. So there are three events that happens: transamination, oxidative deamination, and keto acid modification. So transamination, that first one there, that number one there, many amino acids pretty much can transfer their amine group to an alpha ketoglutaric acid, transforming it into glutamic acid. As a result, the original amino acid becomes a keto acid. Now, the glutamic acid is an intermediate keto acid of the citric acid cycle. But this reaction is reversible. So you can actually like pretty much go back into the amine group. And that's actually transamination, when an amine group is switched from an amino acid into a keto acid. Then you have the second one, which is oxidative deamination. Oxidative deamination is when the amine group of glutamic acid is removed as ammonia and combined with carbon dioxide to form urea. And that happens in the liver. In the liver, the amine group of glutamic acid is removed as ammonia, and alpha ketoglutaric acid is regenerated. Ammonia then combines with carbon dioxide to form urea and water, of course. And urea is released to the blood. And when it gets to be filtered, when the blood gets filtered through the kidney, this urea is excreted in the urine. This urea cycle helps the body get rid of the toxic ammonia that we are actually producing from oxidative deamination. Then you have the third one. The third one is your keto acid modification. The keto acid, this is actually when the keto acids formed during transamination are altered so they can easily enter the citric acid cycle. And keto acid formed from transamination here is altered to produce metabolites that can enter the citric acid cycle. And the major metabolites that are produced could either be your pyruvic acid, which is really like going to enter upon transition reaction, is really going to be entering the citric acid cycle as acetyl coenzyme, in an alpha ketoglutarate acid and oxaloacetic acid, which are all like intermediate products of the citric acid cycle. So glycolysis reactions are reversible. So pyruvic acid metabolites formed can be reconverted back to glucose. And this contributes to another process which is happening in the liver, which is gluconeogenesis, the production of glucose from substances that are non-carbohydrates. And this time you're actually talking about something from the amino acid pool. And let's get to the next slide. So here's a quick summary of the amino acid catabolism. Transamination is a transfer of an amine group from an amino acid to an alpha ketoglutarate acid, generating glutamic acid. And then oxidative deamination removes an amine group as ammonia from glutamic acid and regenerates an alpha ketoglutarate acid. I know that you're probably saying this is too much biochemistry and too much of the big words that you cannot understand if you had not taken biochemistry. But that is the reason why, as an anatomy professor, I always recommend that students take organic chemistry first before they get into AMP, most especially AMP2, because AMP2 has a lot of biochemistry. And this biochemistry, we are not going to be able to explain to you what these words are all about. This is actually in the avenue of learning the semantics of organic chemistry. So I'm hoping that you guys all have backgrounds in organic chemistry so you will understand what this means. I tried giving you like a little bit of a background, what is oxidation and what is reduction, that is in part A. And I'm hoping you guys could actually relate into part B's words as we progress. Okay, then this is probably the last segments in protein metabolism, and that is protein synthesis. Pretty much, this is a process where amino acids pretty much get into, is actually like going to get into um, an anabolic process where um, where it can actually be synthesized into a more what do you call this more a bigger version of itself, which is actually a bigger version of multiple amino acids to form proteins. And there are actually like different types of proteins, hundreds of thousands of proteins, and you have only well, like what, 20 amino acids, but combinations of that according to our genetic code can actually give us different types of proteins. So amino acids are the most important of the anabolic nutrients. They form all proteins as well as a bulk of functional molecules. And protein synthesis that occurs is actually occurring in the ribosomes and is hormonally controlled. For example, the growth hormone, the thyroid hormone, and the sex hormones are playing a major role in stimulating protein synthesis. And synthesis requires a complete set of amino acid for whatever protein that you are producing. If you have all 20 amino acids as a requirement to complete that one protein according to the genetic code, then you must have the number and the complete number and the complete amount for each of the 20 amino acids that is necessary. Essential amino acid must be acquired pretty much in the diet. And so all of those nine essential amino acids you have to make sure is actually carried in your diet. The non-essential ones are actually being produced in the liver. And during our lifetime, depending on our body size, you can synthesize about 225 to about 450 kilograms, about 5,000 to 1,000 pounds of protein in our lifetime. So now we leave now the protein. That was the last macromolecule. Uh, but that doesn't mean that we're not going to mention all of these macromolecules. So we're going to get into the metabolic states of the body. And when we get into the metabolism of the body, got something to do with these macromolecules being either catabolized, broken down, or anabolized being formed in the process. And how do we use this? How the body has to do this, and what effects the will this metabolism be on our health? And therefore, it will also remind us of what the dietary nutrition, or the dietary requirements of the body in order that we can stay healthy. So here's another picture of the amino acid pool, and the dynamic state in which these organic molecules, except for the DNA, are continuously broken down and rebuilt is actually our catabolic anabolic steady state. The body uses a nutrient pool, and for this particular pool that we have here in this picture, it's actually the amino acid pool. So this one stores amino acids, and then if it is actually the general nutrient pool, then it also is a storage for amino acids, carbohydrates, as well as fats. Now, pools are interconvertible because pathways are linked by common intermediates. The amount and direction of conversion are directed by the liver, by the adipose tissue, and by the skeletal muscle. And how this particular pool is actually used by the liver, the adipose tissue, and the skeletal muscle. For instance, your amino acid pool, as in something like here, is actually the body's total supply of free amino acids. And we know that proteins are produced, but they're also lost in the urine, the hair, the skin cells. And they are replaced, amino acid pool is typically replaced by diet. This pool is actually used for as a source for resynthesizing resynthesizing proteins and forming also amino acid derivatives. And then it is also used in gluconeogenesis, and gluconeogenesis is a word that means production or formation of glucose from non-carbohydrate form. And here we are assuming that this is actually gluconeogenesis is actually coming from the amino acid pool. So uh, right here, the amino acid pool is actually like showing you that it's actually being supplied by body protein breakdown when the protein that we have in our body breaks down, and then the dietary digestion of protein, and pretty much is being used in catabolism, body protein synthesis, and synthesis of nitrogen or non-protein compounds. Carbohydrates and fat pools. So let's take a look at this. Instead of just the amino acid, we're done with the amino acid. Carbohydrates and fats are easily interconverted through key intermediates. And two major differences between carbohydrate and fat pools and the original amino acid that we just discussed is that carbohydrates and fats are oxidized directly to produce energy. Amino acid must first be converted to a citric acid cycle keto acid, and excess carbohydrates and fat can be stored as such. Amino acids are not stored, and they are not stored as protein. Proteins cannot be stored. So that's the difference between the two. And also in the what do you call this, in the fed state that lasts for about 4 hours after eating, when absorption of nutrients is occurring, anabolism here would exceed catabolism. This means, guys, that after you have eaten your food and has been broken down into its smallest components and you absorb it, it goes into the bloodstream. The liver would not have anything else done right at that moment, but to take all of these nutrients and pretty much like convert them into the protein or the carbs or the fat that we are that's actually part of our body. Okay, so in this particular state, immediately after we eat and after we have digested the food that we've eaten, anabolism, which is actually the formation of new compounds, would exceed catabolism. Excess nutrients are stored as fats if they are not used, if they're not burned. So for the carbohydrates, glucose is a major cellular energy fuel and can be converted in the liver to glycogen or fat. Glycogen remains in the liver, but fat joins the protein to form the very low density lipoproteins, which is actually the VLDLs, and it is released in the blood for storage by adipose tissue. And we're going to be discussing the faith and where the VLDLs and the different lipoproteins are not on this page, but in succeeding pages. Now, blood-borne glucose would enter the cell, okay, and excess stored would be stored as glycogen, such as this particular pathway right here, where the glucose is stored in the muscle cells as glycogen in the muscles or fat in the adipose tissues. The triglycerides, right around here. Let me see. I don't know if you can see my pointer, but here you go. That's a glucose pathway. They're stored in the muscle as glycogen. And right here, the triglyceride pathway, it is actually stored in the adipose tissue as uh, fat cells. But if it's going to be digested, it's going to be digested into its two components, which is fatty acid and glycerol. So when in the digestion of the triglyceride, it's going to be digested into its smallest particle, which is absorbable particle, and that is as fatty acid and glycerol, which forms chylomicrons from the lymph and are hydrolyzed also to fatty acid and glycerol before passing right through the capillary wall. And I'm hoping that you can remember that in our absorption, digestion of fat that we have discussed in the part A. The lipoprotein lipase, which is a fat hydrolytic enzyme, is the enzyme that catalyzes the hydrolysis of lipids. And this enzyme is very abundant in muscles and in fat tissues. Triglycerides are used by adipose tissue, the liver, and skeletal, as well as cardiac muscle, as the primary energy source. Most glycerol and fatty acids are converted back to triglyceride for storage. So we have our own triglyceride after we digest the triglyceride of the animal source that we have eaten into fatty acid and glycerol. And then our liver would actually be if we're not going to be burning the triglyceride that we take in, then our own liver will be producing triglycerides and will be storing them for our own purpose. And the amino acid, as we know already, and that's actually where the amino acid is, the amino acid, some amino acids, some are absorbed and deaminated in the liver to keto acids that can be used in the citric acid cycle. Okay, there you go. Your amino acid is converted into keto acid. Okay, some are interconverted back into protein. That is actually the protein, the human protein that we have. And some of the keto acids are actually interconverted into fatty acids. And together with glycerol, it's going to be converted back into our own human animal fat, our own animal fat, not the same fat that we have just eaten from, let's say, the chicken. Okay, so excess amino acid, excess amino acids can be stored as fat. So your amino acid cannot be stored as amino acid, can be stored as fat in the liver, and most amino acids are taken up by the cells and used for protein synthesis. Let's take a look at now the absorptive states. This is where after we've taken our meal and has gone through the digestion process and building up anabolically all of the substances in the fed state, we're now going to get into the absorptive state. The absorptive state is controlled primarily by our hormone insulin. Insulin secretion is uh, is actually controlled by the beta cells of the pancreas and is stimulated by blood levels of glucose and amino acids. You have also the intestinal GIP, and GIP stands for glucose dependent insulinotropic peptide. I know that is long, but you got to read this in your textbook. And of course, um, and the parasympathetic stimulation. When insulin binds to membrane receptors, okay, when insulin bind to membrane receptor, let me see if there's a blood insulin, okay, when they bind into membrane receptors, it would facilitate the diffusion of glucose into the muscles and adipose tissues as well. And the brain and the liver take up glucose even without insulin. Insulin is a hypoglycemic hormone that enhances glucose oxidation for energy. Guys, I want you to know that insulin is not only for sugar or not only for controlling the direction, the metabolic direction of sugar. It's also what do you call this, for the production of other um, metabolic products of the body. So insulin is hypoglycemic hormone that would enhance glucose oxidation for energy. It is called hypoglycemic is because when you have a lot of sugar, insulin takes the sugar back into the cells that would pretty much like oxidize the sugar into convert it into energy. And that's actually glucose oxidation for energy. And um, it also enhances glycogen and triglyceride formation. It also promotes active transport of amino acid to the tissue cells. And it also promotes protein synthesis. It also inhibits glucose release from the liver and also gluconeogenesis. This function of the insulin, I am guaranteeing you, will be part of the questions that you will have in your exam. So make sure that you study the purpose of insulin. So insulin directs nearly all events of the absorptive state, and pretty much from what do you call this, uh, coming from the pancreas, that would elevate the production of insulin. The target cells would pretty much like uh, stimulate, be stimulated into breaking down glucose, okay, and It will also increase the active transport of amino acid into the tissues and cells, and pretty much it will elevate protein synthesis. It will also elevate the breakdown of triglycerides, okay, or it enhances the conversion of glucose into fatty acid and glycerol and glycogen. Then what is not shown here is that it it pretty much also stimulates protein synthesis, because protein synthesis is really a very big metabolic thing that you cannot see anymore in this picture. All right, diabetes mellitus. Whoa, that is actually a big thing in the market now, most especially for uh, the American market, where a lot of people are really gaining weight because of the food that is out there. Um, the advertisement of food and how to cook them is all over, and the Americans are taking advantage of it and are gaining weight. Okay, and those fast food uh, markets are not even helping; they're actually like aggravating all of this. And but the bad part is we over um, pretty much we overindulge in food and and the burning of sugar, and a lot of us really develop type 2 diabetes. Diabetes mellitus is inadequate insulin production or abnormal insulin receptors, and it results in the inavailability of glucose to most body cells. So whenever insulin that you are producing is not enough anymore for the cells, you have diabetes, or you're probably producing enough insulin, you got a good amount of insulin, but the receptors in your cells do not recognize the insulin. That's also diabetes. So that will also eventually be affecting your cells not being able to get the insulin because the receptors are bad. So, um, so you have the result will be an excessive high blood glucose levels because you're not taking the glucose and you're not burning the glucose down, and therefore you lose glucose also in the urine. So fats and proteins instead are used for energy instead and can lead to what we call metabolic acidosis, also protein wasting and weight loss. Here's actually your normal route for a good, pretty much like uh, pancreas, where insulin gets into the glucose into the cells, and the insulin receptors of the cells pick up the insulin and would help in the breakdown of glucose. In the type 1 diabetes, there's actually lack of insulin production right here itself, and that is due to the loss of beta cells. The beta cells are actually, it's an autoimmune response. Type 1 diabetes is actually an autoimmune cells where our own cells are actually killing the beta cells of our pancreas, and therefore glucose, it will not anymore be producing glucose, and no glucose gets into the cells. In the same token, in the diabetes too, it could either be like your insulin production is waning down because of age, or there's something wrong with your insulin receptor, so the cells cannot accept insulin like the way they used to. And this usually happens with an aging receptor or an aging cell. So after your um, after your absorptive state, where you have already absorbed the nutrients, you have also what you call the post-absorptive state. Now, the post-absorptive state is actually when uh, you get hungry. Okay, it's also called the fasting state, when the gastrointestinal tract is empty and the energy sources are supplied by breakdown of body reserves, and therefore you are not anymore getting your energy source coming from the food that you're eating. Okay, you're hungry and you're not putting anything in your body, but you have reserve, you have your body fat, and that is actually um, the fasting state. Some people really get into this fasting state as a way of losing weight. They can actually like fast for one whole day. They drink only water. They take very minimal nutrients, and pretty much they let their body reserves, really to take over and um, promote whatever metabolism is necessary for that day with the use of just the reserves and not eating anything. So you have now here you're promoting because you're actually pulling nutrients, your basic nutrients from your own cells, from your own reserve. Then you are looking at catabolism or breakdown of fat, glycogen, which is actually your reserve glucose, and proteins. And pretty much the catabolism here will exceed anabolism. Now, the goal here is to maintain glucose between meals. And so if you have an absorptive state, if you are not really getting into a whole 24-hour fasting state, you have a fasting really in between like your lunch and again a late dinner. After 6 hours, 4 to 6 hours, uh, you will you will actually feel hungry. After 4 to 6 hours of eating your brunch or your lunch, you will feel hungry, and there's not anymore any amino acid pool or anything. Everything has been converted to your own body cells. So you have to pull out some again from this pool and use them for your metabolic thing. What if you are actually still doing work and you do not want to eat, right? Are you going to just drop dead? No, you're not. Okay, so you're pretty much you're going to be picking up glucose and you're going to make it available to the blood when the liver will do what you call glycogenolysis. Glycogenolysis is when the liver converts glycogen, which is actually your stored fat or stored glucose, back into becoming glucose again, so that you will have uh, and glucose is actually the unique molecule that is actually needed to get into the Krebs cycle and produce your own ATP. Remember that without ATP, your muscles will not contract, you cannot do work, or your cells will not be able to do work. You're just going to be dead if you are not going to produce any ATP. And your body will not allow that to happen. You have to produce ATP. So where are you going to get your glucose if you're not eating, if you are in the fasting state in between meals? Well, your body pulls it from the liver, who has the uh, capacity to initially convert excess glucose to glycogen, which is actually the type of molecule where you store glucose. So they're going to chop down again the glycogen and release glucose, making glucose available to the blood. And this fasting state is also promoting the use of fats for energy. Now, glucose sparing saves glucose for organs that need it most, such as the brain. So, pretty much, um, when you break down glycogen, you actually like release again glucose and spares this particular glucose and makes it available, most especially to the brain. We don't think we are not going to be able to do produce ATP, which is actually pretty much needed in these vital organs, the muscle and the brain. And muscle and the brain is the only organs that would need actual glucose. So we need actual glucose to be available for them to produce ATP. So right here in the major events that principal events that is actually pathways to the post-absorptive state, you have catabolism and replacement of fuels in the blood. You have again protein in the body, glycogen in the body, in the liver, and triglycerides in all over the fats in the everywhere. They're going to be broken down again. Triglycerides are going to be broken down into glycerol and fatty acids, glycogen back into glucose, protein into amino acids, and use them use them for the major energy fuels. Glucose, pretty much that is provided by glycogenolysis and gluconeogenesis, and the fatty acids and the ketones are now going to be broken down in the citric acid cycle and the oxidative phosphorylation and release carbon dioxide, water, and the more important fuel, which is ATP. And that's actually part of our very important liver. In the liver metabolism, amino acids are converted to glucose in gluconeogenesis. So even if you do not have any glucose around, for as long as you have your amino acid pool and fat, you can actually produce amino acids. Thereby, if you have amino acids, it gets converted into keto acids and then into glucose. The liver, our friend, our um, unknown hero. A lot of us think that our heart is the only hero. You don't want to lose your heart. You don't want to lose your brain. But the unsung hero of the body is actually the liver. The liver is the most, one of the most productive, if not the most productive organ of our body. And it does so many things in the processing of the food. Remember that when the food has been broken down and absorbed by the body, it doesn't go anywhere in the body first. It has to go via the hepatic portal vein. It is actually going into the liver. And so in the liver does so many things in order to find out exactly if the food that we ate is actually useful. Okay, so um, it will actually like sift in the glucose and convert it into glycogen. So we have now the stored um, glucose in the form of glycogen. And then if we need glucose, we are actually going to, the liver is actually going to do glycogenolysis. This is actually a reserve. Now, glycogenolysis is happening in the liver and it's also happening in the skeletal muscle. Before glucose from the liver is exhausted, if there's actually like going to be like an exhausted uh, process where you are really on extreme fasting or starvation, before glucose from the liver is exhausted, glycogen stores in the skeletal muscles will start to break down. Now, lipolysis in the adipose tissues and the liver pretty much talks about glycerol that is used for gluconeogenesis in the liver. In the catabolism of cellular protein, major sources during prolonged fasting, if you are actually like going on a starvation, um, pretty much the limited amount of protein can be broken down before damage. The amount of fat in the body determines how long a person can survive without food, and that is actually in the post-absorptive state, and that's already like in starvation. So the liver actually, before anything could happen to us, if we are on a starvation mode, the liver will keep us alive. It will release all of the things that we need in order to produce ATP. It will actually break down the glycogen that is actually stored in it, and the muscles. Before the liver loses all of the glycogen, the muscles will actually be breaking down also the glycogen in the muscles. Let's take a look at the hormonal and neural controls of the post-absorptive state. This is actually the post-absorptive is the state where you are getting hungry. So, sympathetic nervous system would interact with several hormones, control the events of the post-absorptive state. More complex, it is actually more complex than the absorptive state, which utilizes one hormone, insulin. The post-absorptive state is triggered by reduced insulin, and release blood glucose levels would also be dropping. Now, glucagon, right here, you have glucagon in your PowerPoint. Glucagon is a hyperglycemic hormone whose release is stimulated by declining blood glucose level, rising amino acids also. So glucagon promotes glycogenolysis. So this is actually your increased plasma glucagon right here. It will stimulate glycogenolysis and gluconeogenesis in the liver. That's actually the purpose of drop in insulin. Lipolysis also happens in the adipose tissues, causing fatty acid and glycerol to be released. And very low carbohydrate meals, such as those people who are trying to lose weight, going on keto diets, and yet starving themselves also. In this particular instant, insulin responds to high amino acid blood levels. If high protein meal is consumed in a keto diet, hypoglycemic conditions may result, which is actually not good for the brain. So you have to really watch out if you're actually going for a high keto diet. Just have to make sure that once in a while you pretty much like give yourself a chance really to get some sugars. Now, uh, under such hypoglycemic condition, glucagon modulates the effect, this hormone glucagon, ensuring adequate blood glucose. Now, the modulation of insulin effects after a high protein and low carbohydrate meal would mean that insulin responds to high amino acid blood level. If high protein meal is consumed, hypoglycemic condition may result, which is very much like just like what I mentioned, not good for the brain, but glucagon is there. So, growth hormone, thyroxine, the sex hormones, and corticosteroids can have an effect also on metabolism. Most have an indirect effect. Here's actually your summary of normal hormonal influence on metabolism, which could help you um, recall or it could help you in your study of the um, hormonal influences on metabolism. And a summary of metabolic functions of the liver, which is actually something that you should really consider looking at because the liver is actually the busiest. Okay, if not one of the busiest of all the organs in the abdominal pelvic cavity. And here is actually a review of what happens to the food that we are eating, the fat most especially coming from the gastrointestinal tract after its digestion. The chylomicrons are received by the lacteals on the lymph, and that's actually where this is. Then the lacteals in the lymph, as chylomicron is now going to be delivered into the blood from the lacteals via the subclavian vein. It will now be delivered into the blood. And right there in the blood, it is now going to pretty much be converted into triglycerides and again into fatty acids in the heart, the muscle, and the adipose tissue. Okay, and then in the what do you call this? In the liver, it's going to be converted into cholesterol and LDL, etc. And I will explain to you what these things are. Okay, let's take a look at some of the metabolic role of the liver. Metabolic roles of the liver will be lipid metabolism, carbohydrate metabolism, and there are some other functions. Okay, you have hepatocytes in the liver, which carry out about 500, about 500 metabolic factors. Just to let you know exactly how busy these hepatocytes are. And the process nearly every class, they will process nearly every class of nutrient. They play a major role in regulating plasma cholesterol levels. They store vitamins and minerals. They metabolize alcohol, drugs, hormones, and bilirubin. And right here, you could see that in the lipid metabolism, the liver is responsible for fatty acid uptake, and it's actually responsible for new lipogenesis or production of new fats and um, production of what do you call this reproduction of old fatty acid, lipid storage, lipolysis, beta oxidation, which I have already explained to you what it is, and secretion of lipids. In the carbohydrate metabolism, it is responsible for the uptake of glucose, and then what is supposed to be done when glucose is plentiful and it's going to be in the liver, then you have to convert the glucose to glycogen, which is actually the stored glucose, the form of the stored glucose. Then if you need again glucose, you have to break it down, and the breakdown of glycogen will be glycogenolysis. And then if you need to form new glucose from other sources other than carbohydrate, you have what you call gluconeogenesis. And then from other factors of protein, what the other functions of the liver will be for protein metabolism, hormone and drug inactivation, and immunological function. And of course, one of the most important thing in digestion, which is actually bile formation. Let's take a look into cholesterol metabolism and regulation of blood cholesterol levels. Cholesterol is not used as an energy source. It is a structural basis for bile salts, steroid hormones, and vitamin D. So cholesterol really is used as a skeletal molecule so you can form, it's actually a structural source only, so you can form bile salts, so you can form steroid hormones like your testosterone and estrogen and progesterone, and of course vitamin D. And cholesterol also is a major component of plasma membranes, and 15% of blood cholesterol is ingested, with the rest that is actually made in the body, primarily by the liver. So you have, you do not really need, we do not really need to eat substances that's high in cholesterol. As a matter of fact, uh, I would suggest avoid it, so it's not going to pretty much like increase your blood cholesterol level. But somehow, if you are really into producing more cholesterol and it is in your DNA, there's nothing much what you could do with your diet. What you can do is really to pretty much like take in statins, or take in a medication that is going to be prescribed by your doctor that will um, that will actually slow down your liver's production of cholesterol. Okay, because there are people who really produce only the normal amount of cholesterol that is needed, but there are people who actually produces cholesterol much more than what they can actually process. And you don't really need too much cholesterol if you're not really, most especially the older people, will not anymore be producing those hormones. So you're producing too much cholesterol is not anymore necessary. So cholesterol is lost from the body, though, when catabolized or secreted in the bile salts that are lost in the feces. Okay. So here you go. Your cholesterol is actually like produced from your acetyl coenzyme. It's now and stored in the liver. It's now going to be um, pretty much like um, converted into bile salts. Okay, and pretty much this bile salt is actually a necessary uh, component on the digestive process, and it's an emulsifier. Then some of the cholesterol is actually also going to be converted into your VLDL, very low density lipoprotein, and your low density lipoprotein, and your high density lipoprotein for some of it, very little of it though, very little cholesterol. So you have cholesterol, most especially in the LDL, which you do not want to happen. It gets into circulation. Okay. So what are lipoproteins? I have mentioned to you they become cholesterol becomes part of the LDL and the VLDL. So what are they? Okay. Lipoproteins are transport water soluble cholesterols and triglycerides through the blood. So these are transporters of cholesterols and animal fat. They regulate lipid entry and exit at the target cells. They all contain triglycerides, meaning all of the lipoproteins. You have your very low density lipoprotein, your low density lipoprotein, and high density lipoprotein. They all contain triglycerides, phospholipids, and, and cholesterol, as well as protein. And the higher the percentage of lipids, the lower the density. So there are different types of transport lipoproteins. The first one is what you call the high density lipoprotein, this one here. It has the highest protein content. They will transport excess cholesterol from the peripheral tissues to the liver. And then it can be broken down and secreted into the bile. It also provides cholesterol to steroid producing organs. HDL is thought to be very protective because of its role. It's very protective against heart disease because it takes cholesterol out of the blood. So if you have a good amount of HDL in your body, then you are good because of the role of the HDL and what it does to the body. It is important that you increase the level of HDL in your body. And 60, uh, the level of HDL 60 and greater than 60 is good. Lesser than 40 is not very good. So keep your LDL high. Well, you know, the omega-3 and omega-6, the fish oils, are actually like very rich in HDL. And taking this oils, fish oils, is actually going to help you increase the level of your LDL, okay, HDL rather. So LDL, let's now go to the low density lipoprotein that carries cholesterol from the liver to the tissues. The LDL has the highest cholesterol content. They transport cholesterol to the peripheral tissues for membrane storage or hormone synthesis. LDL cholesterol deposits cholesterol also in blood vessels, which is really not good. 100 or less is good for your LDL level. 130 or above is not good. So you have to minimize the level of LDL. In your blood serum, in your blood test, your LDL level must be lower than 100 to stay healthy. Now your VLDL, very low density lipoprotein. This contains more than half triglycerides with low density of proteins. They transport triglycerides from the liver to the peripheral tissues, mostly adipose tissues. And pretty much, it's not really very good. Chylomicrons, which is actually the digested form of the fats transported from the gut, have the lowest density and consist almost entirely of triglyceride. The maximum total cholesterol that you have, according to the list, if you are going to get into what the serum level of this you should have, the maximum total cholesterol that you must have will be 200 milligrams per dilution or less. Okay, 200 milligrams per dilution is already linked to atherosclerosis. So if you could go lower than that, that would be good. Most important to measure form in which cholesterol that is actually transported in the blood, which is chylomicrons. So what are the factors affecting uh, or regulating the blood cholesterol level? Hmm. There's actually a lot here, right here. This is the easiest one that I could think of to get my students to understand more about this. The liver produces cholesterol at a basal level, regardless of dietary cholesterol intake. Uh, Restricting the dietary cholesterol does not markedly reduce blood cholesterol level. If it is in your DNA to produce a lot of cholesterol, there's nothing much you could do except take in medications from prescribed from your doctor so that it can stop your liver from producing too much cholesterol. The more important effect is the relative amount of saturated and unsaturated fatty acids. So saturated fatty acids stimulates the liver to synthesize more of cholesterol and inhibit cholesterol excretion from the body. And that's why an increase in saturated fatty acids is not good. So you don't want to be eating food that is rich in saturated fat. Unsaturated fatty acid, on the other hand, those fatty acid that polyunsaturated, monounsaturated, okay? These are the unsaturated fatty acids. They enhance the excretion of cholesterol into bile salts. So the more unsaturated fatty acids that you get, uh, more of those avocado oil, or peanut oil, or um, um, grapeseed oil, or the monounsaturated, so whatever unsaturated that you have, they would enhance excretion of cholesterol into the bile salts. Trans fats. Do I have trans fats here? All right, right there. Trans fats. Trans fats can occur when healthy oils are chemically transformed into solids. An example of this is your margarine. Your margarine has the worst effect on cholesterol level than saturated fat. It increases LDL and reduces the HDL. So if you can avoid margarine as much as possible, avoid it. Unsaturated omega-3 fatty acid that is found in cold water fish have lower proportions of saturated fats and cholesterol, and pretty much is one of the healthiest nutrients that you could actually take in. They make platelets less sticky and help prevent spontaneous clotting. They have an anti-arrhythmic effect on the heart, and it can lower blood pressure. On the contrary, stress and cigarette smoking had been shown to lower the HDL, which is not good, right? Uh, well, aerobic exercises and estrogen increase HDL levels. Well, you want your HDL to be lower, I'm sorry. But cigarette smoking, stress and cigarette smoking lowers HDL levels. Oh, that means it's good, huh? Aerobic exercise and estrogen increase HDL levels and decrease the LDL levels. Now you're probably thinking, well, what about body shape? The shape of my grandma is changing. Well, that is something that we cannot avoid. Some of us will lead to becoming like will be forming a lot of fat around our belly and will look like an apple. The fat that is actually carried on the upper body is correlated with high cholesterol and LDL levels. The pear fat, where your fat is actually like carried on the hips and the thighs, is correlated with lower cholesterol and LDL levels. Lower cholesterol is really good. Huh. Anyway. Okay. The risk factor for assessment of cardiovascular disease pretty much uh, will be the previously high cholesterol and LDL to HDL ratios. And these were used as predictors of potential disease and was found not to be as reliable. Now LDL levels and other risk factors are believed to be more accurate. The treatment for high LDL levels actually is your statins. Statins are cholesterol lowering drugs estimated at about 10 million Americans that's actually under this regimen. A lot of people have this problem and would like to lower cholesterol and triglycerides. So, you are at risk when your cholesterol level is 200 to 239. Remember that what do you call this? The healthy cholesterol level should be at 200 and lower. If you go beyond that, up to 239, you are at risk. And your LDL should be lower than 100. And anything that is actually over than 100, you are at risk. And your HDL, you need to have a higher rather, about 40 to 59 in a male and then 50 to 59 in a female. Why do we need more? Okay. So when your cholesterol level is 240 and higher, you're already at the dangerous level. LDL cholesterol, that is 160 and higher, is also dangerous. You have to keep it low, as a matter of fact, lower than 100. And HDL and cholesterol for males, 40 for male is dangerous, and for 50 in a female, under 50 is actually dangerous. So you need to keep your HDL high. Now, what will be a healthy person? When your cholesterol is under 200, when your LDL is under 100, and your HDL is actually higher than 60, your high density lipoprotein is actually good. Remember, guys. Now, when people say, well, uh, my grandpa died 93 and he never stopped smoking. And you know that smoking is actually going to uh, pretty much like increase your LDL. And that is not good. You know, I was kidding when I was saying that is good. No, that is not good. And um, then that person who pretty much like lived up to 93 and was a smoker, and you know that cholesterol and LDL is actually being promoted by smoking itself, then genetically they probably have the capability of producing less cholesterol. And they are fortunate to have a liver like that. But not everybody, even if it is actually familial, even if you tell everybody that your grandpa died at 93 and is drinking and is actually smoking and is actually um, eating fatty food and triglyceride at the age of 93, good for him because he has a very good liver. But not everybody down the line. You could be inheriting, DNA um, from your grandma or from your parents, from your parents that is not actually directly from your grandpa. And too bad for you, you might end up dying earlier. So make sure that you are aware of when you are at risk and stay healthy by making sure that your cholesterol is under 200, your low density cholesterol is under 100, and then your HDL cholesterol is 60 and higher. Keep it high. Okay, let's take a look now at energy balance. The bond, the bond of energy released from food must equal also the total energy output. So if you're getting energy from food and you are active daily, you must also make sure that all the energy that you got from food is what you're going to have as your output. Your energy intake is the energy liberated during food oxidation. Energy output includes uh, energy that is immediately lost as heat, as body heat, which is about 60%, and energy that is used to for us to do work, which is driven by ATP, and energy that is stored as fat or glycogen. Nearly all energy from food is eventually converted to heat, which cannot be used to do work. Now, heat energy pretty much is only there to warm tissues and blood, and it helps to maintain homeostatic body temperature. It allows metabolic reaction to occur. Temperature has got something to do with that for a metabolic reaction to occur efficiently. If energy intake equals energy output, then a person's weight is stable. If it is not equal, then there will be either a gain, okay, if you're not burning what you're eating, or there could be a loss if you're actually burning too much more than what you're actually eating. So let's take a look at BMI. What is basal metabolic index? Basal metabolic index is a formula used to determine obesity based on a person's weight relative to height. And pretty much with me, I know I fall under 27, which is a class 1 obesity. So I am obese. A person is considered overweight if the BMI is about 25 to 30. Overweight. I am overweight, and that's actually on my record. And obese if the BMI is greater than 30. Obese people have a higher incidence of atherosclerosis, type 2 diabetes mellitus, hypertension, heart disease, and osteoarthritis. And pretty much, guys, I'm already getting into that stage, and I am getting into all of those symptoms. Hi. Yeah, yeah, yeah. The current statistics, though, is that 2 out of 3 adults are overweight, and 1 out of 3 is actually obese, and 1 out of 10 has diabetes, and 17% of children, and we're talking here of the new generation, okay, is already obese, which actually, if you compare it with years ago, probably during my time, only 5%, about 40 years ago, or that's actually like when I was young, was only 5%. I feel so bad, really. Using this particular formula here, where the body mass index is equal to your weight in pounds times 700, okay, and the square of your height in inches. First thing to do would be to look for your height in inches, multiply it at the same time square, okay? Then you divide 705 with that, multiply it with your weight in pounds. Then you're going to get your body mass index. My mass index is about 26.8, so almost 27. I am not obese yet, but class 1 obesity. I am overweight. Okay, let's take a look at pretty much like the control of uh, energy and expenditure and hunger. Uh, feeding Our feeding behavior and hunger is regulated by neural signs from the digestive tract. This is actually your digestive tract, okay, and pretty much these blood-borne signals is related to uh, energy stores. It is also getting signals from hormones in the body, and then to a lesser extent, body temperature and psychological factors. Now, They all operate through the brain thermoreceptors and chemoreceptors and pretty much others. Food intake is subject to both short and long-term controls. So pretty much our push to eat, okay, is actually being controlled by so many factors from hormones to our body temperature and our environment. There are areas in our hypothalamus that would release peptides that would influence our feeding behavior. Our hypothalamus is the seat of hunger and satiety. It is actually the one that will make us feel hunger and will make us feel also satisfied. Our hypothalamus has the ability to do that. And you're probably going to say, I thought it was the digestive system. Well, the digestive system does the mechanics of digestion and absorption of the food that we take in. But the controlling mechanism for when we're going to be taking in food is actually controlled by hormones as well as uh, the hypothalamus, which is the seat of hunger and satiety. Hormones, gut hormones, such as your insulin and your cholecystokinin, can actually depress hunger. Remember what I mentioned during your digestion in your stomach? Okay, whenever the first drop of chyme enters the duodenum, that's going to, cholecystokinin is going to be released by the liver right through the bile and secretin, and both of these uh, hormones are going to depress hunger. It will slowly inhibit gastrin. It will inhibit the motility of the stomach, and therefore you feel that you are already full. Glucagon and epinephrine, on the other hand, will stimulate hunger. Ghrelin from the stomach, it's actually a hormone from the stomach, stimulates the appetite. The levels usually will peak about a few minutes before mealtime. There's also long-term regulation of food intake. For instance, let's take a look at leptin. Leptin is a hormone secreted, okay, right here, okay, leptin. It's a hormone that is secreted by fat cells in response to increased body fat mass. It's an indicator of total energy stores in the fat tissue. Leptin protects against weight loss in times of nutritional deprivation. It acts on the ARC neurons in the hypothalamus. It suppresses secretion of the NPY, which is a potent appetite stimulant. It stimulates the expression of appetite suppressants. For instance, your peptides, which you call the CART peptides. And there are other factors besides leptin that regulates your food intake. Temperature, your cold activates hunger. Then you have stress, depending on the individual. Usually, if you're under stress, you do not have the appetite, okay? But there are some people who become really overly stressed in the point of psychosis that would overeat, and they will do nothing but binge on food because of the stress. Now that is actually not normal. It's actually a form of binging. Is a form of psychosis, and you have all and it becomes a psychological factor. Adenovirus also, and sleep deprivation, and pretty much some composition of your gut bacteria. And rising leptin level causes some weight loss, but is no magic bullet for people who are really very obese. Obese people have high leptin levels, but seem to be resistant to this action. You know, you're supposed to lose weight when you have high leptin levels, but it was noted in a lot of studies that there are obese people that have high leptin levels. So at this point in time, these obese people have pretty much the receptors of their cells seem to be resistant to the action of leptin. The theory on the function of leptin is that it helps prevent weight loss during times of nutritional deficiency, period. But there are also some acceptable um, inconsistencies, most especially for people whose cells do not recognize the function of leptin. Okay, so even if you have leptin out there, if your cells are not accepting any information from leptin, then how could leptin um, do? How can leptin help you lose weight? There are also some short-term regulation of food intake. There are some neural signals from the digestive tract. For instance, you have already, like, if you have eaten something that is high in protein, the high protein content of your meal would increase and prolong the afferent vagal signals. And these tensions would send signals along the vagus nerve that suppresses the hunger center. Therefore, eating a lot of protein will make you go a long way. Eating less of the protein will make you immediately hungry. And that's the reason why eating pasta or rice, which are all carbohydrates, will make you hungry um, a shorter period of time. But protein, pretty much the neural signals from the digestive tract, if your meal is actually high in protein, would increase. Now, nutrient signals that is related to energy stores increased nutrient levels in the blood would depress eating. So for as long as you've just eaten and you have just like digested your food and you have a lot of nutrients that is in the blood and it's in the liver, you will not feel hungry. Okay, you will have you have high glucose level immediately after a meal. You have elevated amino acid level immediately after a meal, and your blood levels of fatty acid is high, so there's no need really for you to be hungry. There are also other factors that regulate food intake. Temperature, just like what I mentioned, cold activates hunger. uh, there are some what do you call this, other factors. But right here, um, got something to do with social cultural practices. Some people pretty much could, um, who will have the habit or has the habit really of going into fasting because of some social cultural practice or even religious practice. Okay, and there's also like the age-based food response. The response that we have into eating food, we carry it all the way to our aging. We think that this is how much we are used to eating, so I will be eating the same amount of food. But wrong again, we are not anymore going to be metabolizing as fast as when we are younger. So pretty much the age-based response should take into consideration that aging will slow down metabolism, and should your response to food intake must also be lower. And of course, there are some functional food claims that people understand and would follow. They would like really to go on a keto diet because of the protein. They would like to go into less into a carbohydrate diet because of the glucose in it. Well, got something to do also with food preferences and portion size. And I guess if your stomach is actually used to big portions, your body is always going to look for, your stomach is always going to look for big portions. But if you keep your portions smaller, you get it to be like smaller and smaller, you find out that um, your habit will actually be also eating small portions. Then you have your moods and food cravings, which some people will find really very hard to control. And genetics, which is actually like uh, something that we cannot really like suppress. Like if you have the genetics really to produce a lot of cholesterol, there's nothing much you could do but take medications, right? And of course, regional variations, which is wherever you came from, the food that you're eating actually goes with you. You grew up eating rice, and you transfer to another place, even if rice is not the staple there, right? You will be looking for rice. And gender differences, men and women really pretty much eat differently. And for instance, I love vegetables, but my husband loves meat. So pretty much this is actually our gender difference. And I know that from among our friends, there are a lot of females who are aging to love more vegetables as they're aging. And the men doesn't change. If they love meat when they're young, they still love meat because they still have their hormones, and pretty much are they are not linking their eating habits to the loss of any hormone because they still have their hormones. And the females, whenever we're aging, we lose already our hormones, and I guess somehow uh, that changes everything, including the way you eat. And of course, our sleep, our physical activity, and our obsogenic environment pretty much will affect us, our eating habits. So, what is meant by basal metabolic rate? Basal metabolic rate reflects the energy the body needs to perform its most essential activity. So, it is measured in the post-absorptive state, which means after the absorption, 12 hours fasting, in a reclining position, relaxed, you're relaxed mentally and physically at room temperature that's about 20 to 25 degrees Celsius. It is recorded as kilocalories per square meter of body surface. For instance, your 70 kilogram adult basal metabolic rate would be 66 kilocalories per hour. And basal metabolic rate is influenced by age and gender. Basal metabolic rate would decrease with age, and the males have disproportionately higher BMR because of their testosterone. Body temperatures is also affecting BMR. BMR increases with temperature. And stress also. Stress, BMR increases with stress. And thyroxine, which is actually a production of your thyroid gland, increases oxygen consumption, cellular respiration, as well as your BMR. So the basal metabolic rate can be measured directly by a calorie meter, and it measures heat liberated into a water chamber, and indirectly also via a respiratometer or respirometer. It measures oxygen consumption directly proportional to heat production. So your basal metabolic rate is actually the energy that equals the energy that is equaling energy in to energy out. Okay, so that's actually your basal metabolic rate. It reflects the energy body needs to perform its most essential activity. Okay, let's take a look at some homeostatic imbalance. We have here hyperthyroidism. Hyperthyroidism, that means an increase in the production of thyroid hormone by the thyroid gland. And it causes many problems resulting from high basal metabolic rate it produces. The body would catabolize stored fats and tissue proteins despite increased in hunger and food intake. The person often would lose weight, but the bones will weaken and the muscles, including the heart, begin to atrophy. Hypothyroidism, on the other hand, would result in slowed metabolism. So it would cause eventually obesity and diminished thought process. The total metabolic rate is the rate of kilocalorie consumption to fuel all the ongoing activity. Total metabolic rate increases with skeletal muscle activity and food ingestion. This is what you call food induced thermogenesis. It is the greatest with protein and alcohol ingestion. Now, hyperthyroidism causes many problems resulting from high BMR. Okay, and it is when the body catabolizes stored fat and tissues, and that's why that person with hyperthyroidism would lose weight. Now, uh, let's take a look at regulation of body temperature. The normal body temperature is 37 degrees Celsius, which is about 5 plus or minus in degrees Celsius, or 98.6 degrees Fahrenheit. There is an optimal enzyme activity within this temperature in our body. This is our optimal temperature for optimal enzyme activity. So if our body temperature increases, if we're having fever and the body temperature increases beyond 98.6, let's say it increased to 100 degrees Fahrenheit, then you lose the optimum enzyme activity temperature. So this increased temperature will denature some proteins and would depress the activity of neurons. In children under 5, a temperature of 41 degrees Celsius or about 106, that's already too much, right? Can lead to convulsions. And about 43, which is 109 degrees Fahrenheit, is the limit for life. We can die if there's already like 109 degrees Fahrenheit, which means our enzymes would not work, period. So tissues can actually tolerate lower body temperature better than a higher body temperature. That's why when a person is in experiencing a high fever, doctors really would do everything to lower down the body temperature and pretty much put the body temperature within the normal range for all of our enzymes to work properly. And that's the reason why we feel sick if our body temperature go up, because our enzymes are not working the way there should be. Some of them are slowing down, some of them are stopping completely. And when these enzymes, which happens to be the substances that would increase metabolism, okay, would speed up metabolism, is not working, then our metabolism will slow down. And we are not going to be able to do things cellularly, intracellularly, that our body needs to do. And when things do not really happen the way it should be, then that's when we feel sick. So this simply means that the body temperature reflects balance between heat production and heat loss. At rest, the liver, the heart, the brain, the kidneys, and endocrine organs generate most of the heat. And then during exercise, heat production from the skeletal muscles would increase dramatically. And when this heat production while we're exercising would dramatically increase, what's going to happen? Is the body just going to allow all of this heat produced during our uh, exercise, during the process of exercising? Would the body just let it increase inside our body? No, we are going to make sure that something happens to release the heat, and that's when we sweat. Sweating is a homeostatic response to lower down body temperature. We lose water with the heat because too much heat is not good for um, the optimum temperature that we need for enzyme function. So body temperature remains constant for as long as heat production and heat loss are balanced, then your enzyme activity is going to be perfect. There are four mechanisms for transfer of heat that is actually occurring. Number one is, well, first of all, let's try to look at this picture right here. The core organs within the skull and the thoracic and abdominal cavities has the highest temperature, and rectal temperature is best at best is a clinical indicator. Core temperature is regulated and is fairly constant. Blood is a major agent of heat exchange between the core as well as the shell. The shell that we call is actually your skin, and it has the lowest temperature. It fluctuates between 20 degrees Celsius to 40 degrees Celsius. So let me go back to the mechanism of heat transfer. It's actually like four. I mentioned that. Radiation is a loss of heat by infrared rays. Conduction is a transfer of heat by direct contact. Convection is a transfer of heat to the surrounding air. And evaporation is heat loss due to the evaporation of water from the body surfaces. And heat absorbed by water during evaporation is known as the heat of vaporization. There is also what we call insensible heat loss. Insensible heat loss accompanies insensible water loss from the lungs, the oral mucosa, and the skin. A loss of about 10% of basal heat production is actually insensible heat loss. It's lost through insensible heat loss. Sensible heat loss, on the other hand, occurs when the body temperature rises and sweating increases, and sweating would increase water vaporization. So, what pretty much controls our changes, the temperature changes in our body? Well, the temperature changes in our body is actually regulated by the thermoregulatory centers, which is in the brain. It's actually the preoptic region of the hypothalamus, and that is considered to be the main integrating center for thermoregulation. I will say that again. Preoptic region of the hypothalamus is the main integrating center for, that's right there. I cannot pretty much like point to you where because it's a very small drawing. So there are two regulatory centers. One is a heat loss center, and the other one is the heat promoting center. And the heat loss, pretty much the heat generation is the heat promoting center, and the temperature reduction is a heat loss. The hypothalamus receives afferent input from the skin and in the body itself, from the peripheral thermoreceptors in the shell and central thermoreceptors in the core. And pretty much this is actually how the hypothalamus would receive signals about our changes in body temperature. Then it would initiate the proper heat loss and heat promoting activities. And uh, for instance, in the temperature reduction mechanism to reduce the temperature in the body, then your sweat glands will actually be activated, and therefore you're going to lose heat through sweating. There's going to be vasodilation of the smooth muscle of the blood vessels, and pretty much it's going to help you move the water, the blood itself, into the skin in order that there's going to be um, sweating and loss of temperature. Now, with the generation of temperature, that's increased temperature in the body, then this is going to be stimulation of the adipose tissues as well as the somatic muscles. And for immediate production of heat, the only thing you need will be to make the only thing that the hypothalamus will do will be to make your muscles like contract, as in the shivering, and that will affect the somatic motor neurons, and this will already produce body heat. And in the adipose tissues, these brown adipose tissues are actually the ones that are producing heat energy for us. So pretty much these, all of this, the will actually increase the metabolic uh, increases in metabolic rates are pretty much are brought about by, as in for instance in the shivering effect, rather in the non-shivering, the non-shivering response here is actually by chemical thermogenesis via epinephrine and norepinephrine that is stimulated by cold temperature. And the brown adipose tissues in infants and adults is pretty much a breakdown of this adipose tissue is actually thermogenic. It's actually a thermogenic reaction. Okay, this is non-shivering. The other one is shivering, which is actually brought about by the action of the skeletal muscles. And then you have also the enhanced release, which is not here, the enhanced release of thyroxine, which is a secretion of the thyroid gland. It is only seen in infants. And of course, some behavioral mechanisms that is actually seen in adults, such as putting on more clothing, drinking hot fluids, changing posture, okay, so that our temperature is controlled. Increasing physical activity if we want thermogenesis, or decreasing physical activity we want to um, if we want to be a little bit cooler, or staying inside the house if you don't want thermogenesis, and staying outside if it's hot, if you want more of the heat. So right here is actually right here is mechanisms of body temperature regulation, and this is actually in your textbook. Okay, let's take a look at how this thing is actually going. First of all, you will have your stimulus, which is increased body temperature. The blood becomes warmer than the hypothalamus at a set point, which means you're actually increasing your body temperature, which is not really good. There's an imbalance here. The normal body temperature is 35 degrees Celsius to up to 38. Okay, so that's actually what we want to achieve. But then there's an increase in body temperature. So these would activate a heat loss center in the hypothalamus. And that's going to make you sweat. Your sweat glands would secrete sweat, which is vaporized by the body, helping to cool the body. The skin and the blood vessels will dilate, capillaries will become flush with warm blood. These will promote the heat to be radiated from the skin surface. And then eventually, the effect will be body temperature decreases, blood temperature declines, and the hypothalamus heat loss center shuts off. That's actually balancing it. What about if the stimulus is decreased body temperature? Here, this time you're actually experiencing hypothermia. What will be the body's initial reaction? First, it will activate the heat promoting center in the hypothalamus. And then your skin blood vessels will constrict, which will divert blood from the skin capillaries to deeper tissues, minimizing overall heat loss from the skin surface. And your skeletal muscles, right here, okay, is activated when more heat must be generated. This is when shivering will occur. And eventually, after having all that, the body temperature increases, the blood temperature rises, and the hypothalamus heat promoting center shuts down. So, fever is a hyperthermia, mostly due to infection, but also cancer, allergies, or central nervous system injuries. The macrophages release cytokines called pyrogens that cause release of prostaglandins from the hypothalamus. resetting our natural thermostat on a higher level. Fever pretty much triggers heat producing mechanisms, and the temperature is becoming higher. Now, natural body defenses or antibiotics would reverse the disease process if you are sick. Cryogens, for example, your vasopressin, reset the thermostat to lower the level, activating heat loss mechanism so the temperature will actually fall. You have to make sure that you know that there are some medications that you can take if you have fever that will pretty much like affect uh, actually what you call cryogens that will affect and reset our thermostat to lower our body temperature. And that is the end of chapter 24. I will see you next week in chapter 25.